Hello everyone, my name is Rahul. In the previous video, we had discussed about isolation of DNA. In this video, we are going to continue with our discussion on processes of recombinant DNA technology, and we will be discussing about fragmentation of DNA. Now, fragmentation of DNA or DNA cutting, it is performed by incubating the DNA with restriction endonucleases at optimum conditions for a particular enzyme. Each restriction endonuclease will have different temperature condition, will require different temperature, different pH for its optimum action. We already know about the temperature and condition requirements for various endonucleases by experimental means. So whatever type of endonuclease we are using, we will incubate the DNA with that endonuclease at the optimum conditions which are required for that endonuclease. Now, after the addition of restriction endonuclease, the DNA, which is present as a long molecule, it will be fragmented into a number of smaller fragments. Now, not all of these fragments will be of use to us, only a certain fragment will be of use. We will be knowing the size of that particular fragment by spectroscopic analysis. We would have already analyzed the size of the required gene of the required fragment. So what we will do is to separate these fragments and to obtain the fragment of our interest, we will perform a technique known as agarose gel electrophoresis. In agarose gel electrophoresis, we take a plate made up of agarose gel and we make some depressions which are known as wells. So we will add our DNA, we will add all our fragments into these wells. And we will pass electrical current. This will be the negative electrode, this will be the positive electrode. Now we know that DNA molecules are negatively charged particles, so they will move towards the positive electrode. Now this movement depends upon size. Movement in agarose gel depends upon size of the fragment. It does not depend upon the charge. The fragments will have nearly the same charges, but rather the movement in the agarose gel depends upon the size. So the smallest fragment will move the farthest and the largest fragment will move the least. So smallest fragment will cover the most distance, and the largest will cover the least distance. Now by our analysis, we will already be knowing that what is the size of our DNA fragment, and we will know how much that size will move. So for example, if we had already known that our the required DNA fragment will move this much, so these will be our fragments of interest. These will be the fragments that are our genes that we will be using for our further procedures. So this is known as agarose gel electrophoresis. After performing agarose gel electrophoresis, we will have to visualize these DNA fragments. These DNA fragments can only be visualized. DNA fragments can only be visualized by staining with a dye called as ethidium bromide. ethidium bromide. So we will stain the fragments with ethidium bromide, and when UV light will be shown upon the fragments, they will give a bright orange color. They will give a bright orange color. So bright orange color in the presence of ethidium bromide stained gel. Now these separated fragments, they are cut. These separated fragments, they are cut out of agarose gel. They are cut out of agarose gel and placed on nitrocellulose papers. Then they are stained with ethidium bromide. Then UV light is shown and bright orange color is obtained, and we will know which is our fragment of interest. So the DNA fragments purified in this way, they are used in constructing recombinant DNA by joining them with cloning vectors, which we will see in the next video. This method, this process of cutting out agarose gel fragments and placing them on nitrocellulose papers, this is known as elution. This is known as elution. So with this, we finish off with our discussion on fragmentation of DNA. See you in next video.